of a seed. Okay, so now we discuss about germination. So what is germination? Germination is a process of formation of small plantlet from a seed. This is the simple definition of germination. What is germination? Is the process by which a small plantlet will form from a seed. Okay, so generally seeds are in dormant stage. What is dormancy? Dormant stage means inactive stage. The same pea seed we can take example. When generally you can see mothers are storing pea in a bottle or in a jar, and they are keeping it in a storeroom or in a cupboard anywhere. So at that time that pea seed is in dormant stage. It will not germinate into root and shoot. But if we will take the same pea seed and if you will put it inside ground, you will provide water to it, okay, you will provide adequate temperature to it, then that pea seed will germinate into root and shoot. Okay, so when seeds are in inactive stage, means when they are not producing root and shoot, that time we call a seed as dormant seed, or we call that stage as dormant stage. But when that dormant seeds will get adequate environment for their growth, they will develop into what? A new plant. So germination is a process of development of plantlet, or you can say development of a plant from a dormant seed, or simply you can write from a seed. So already we have discussed about the parts of a monocot seed and a dicot seed. In our parts of seed, in that video already we have discussed. So today I will show you how these monocot seed and the dicot seeds they will germinate. Okay, so here for both I am showing you. Suppose, okay, this side I am doing for monocots. Okay, this is the soil. Okay, and this side I am doing for dicots. Okay, so here I am writing monocot seed I am writing, and here this I am writing dicot seed. Okay, so when we will put a monocot seed inside the soil, okay, we will put a monocot seed inside the soil. Here I am putting the dicot seed, like this. Remember, in our parts of seed portion, I have done the two parts or two cotyledon of a dicot seed. Already at that time also I said the seed is in like this format. Okay, in closed format. There for showing the parts of the seed, I have shown in this format. Okay, by open format. Okay, so here this is the dicot seed and this is the monocot seed. So already we have discussed about the plumule and radicals. Here I am doing, this is the embryo. Okay, and here also it contains the embryo. Okay, it got plumule and radical. Okay, it also got plumule and radical. Okay, so these plumules they will develop shoot, and these radicals they will develop into shoot. Okay, so when we will put these seed inside soil and we will provide water. So one more condition I am writing. First I am writing water. Okay, water is a very essential or very necessary thing for the germination of seed. Why? Because when already we have discussed about micropyle ant or micropyle pore. Okay, so when we will apply water, the micropyle pore of the seed they will absorb water, and after absorbing water, the seed coat will burst. Okay, the seed coat, the covering of the seed will burst, and the plumule and the radical they will emerge. Okay, that's why water is a very essential element for the development of or for germination. Okay, so here when these seeds they will absorb water, they will develop into, here I am saying. So and the radical, they will develop a tiny root. Okay, here also I am showing you these seed. Okay, here like this only. Okay, this is the plumule and this is the radicle. Here I am writing. This part is only the plumule. Okay, like see, like this it is in dicot and like this it is in monocot, and this is the radicle. Okay, here this is the what plumule and this is the radicle. This is the radicle. Okay, so after developing a small or tiny plumule and radicle, again there is development. What development? It will come out from the soil. Okay, it will come out from the soil. Okay, the same here. See how here it will come out. Like this it will come out. Okay, can you find the difference? Yes, here the seed along with the cotyledon, they are inside the soil. Only the plumule emerging out from the soil and the radicle, they are emerging down to the soil. But here you can observe the seed along with the cotyledon. Cotyledon is just present after endosperm, I said. So here the seed along with the cotyledon, it will come out from the seed. Only the root or the radicle will be inside the soil. Find the difference. Okay. Then after that, okay, will form a new plant. Suppose a maize. Okay, here also it will fully come out. And now the cotyledon will be detached. This is the cotyledon. Okay, this cotyledon will be detached from the seed, and then both the cotyledon and the seed, they will get dry and come out from the plant, only remaining the shoot. Only remaining the shoot. This cotyledon, it will dry and it will fall down. Again I am repeating. So here you can see the development phase of seed. We call it as the phase of germination. This is the phase of germination for monocot and this is the phase of germination for dicot. So here we have taken a monocot seed or a maize seed, any monocot seed we can take. Here we have taken any, where we can take any dicot seed. Okay. So here we can see the phase of development for both monocot seed and for dicot seed. For monocot seed, you can see the plumules and the radicals, they will develop. You can find the difference how the plumule and radicals they are developing in monocot seed and how the plumules and radicals they are developing in dicot seed. Okay, if possible, I will show you also the germination of monocot seed and dicot seed in practical format. Okay, so that it will be very clear. We will put a maize seed inside soil and we will put any dicot seed inside soil. Then we will see the result, how it is germinating in a soil. Okay, so you can do this by your own also. Okay, in your garden, you take a small portion of soil, then you put inside you put a small monocot seed and you put a small dicot seed. Then find the difference, how they are germinating. in monocot seeds, the seed will remain inside the soil. Only the shoot and the root will come. The root will go inside the ground and the shoot will come out. But in dicot seeds, the seed along with the cotyledon, it will come out. Okay, here the seed along with the cotyledon will remain inside the soil. Okay, but here the seed along with the cotyledon, it will come out from the soil, and at last when the shoot will develop, the seed and the cotyledon, it will get dry and it will fall down. Okay, so this is the what development phase, or this is the germination phase for both monocot seed and dicot seed. Okay, so when we are talking about germination, there are some conditions that are necessary for germination. The first condition already I said, water. Okay, water is a very essential or very necessary condition that is required for the germination of seed. As I said, when the micropyle end or the micropyle, it will absorb water, the seed coat will burst, and it will be very easy for the seed to produce the plumule and radicle out from the embryo. Okay, now the second condition we can say oxygen. Okay, or air. Okay, so these oxygen, they are absorbed by the seed. Generally you can see the soil which are very tightly packed, that kind of soil will not give better yield or better production. The soil should be porous. That's why the farmers, they are digging the soil in regular interval so that the seed can get adequate amount of oxygen. We call that kind of soil as aerated soil or porous soil which contains pore over it. Okay, so that's why in flower vase or in our garden, we should dig the soil in regular interval so that the roots or the seed, they can get adequate amount of air. So oxygen is also playing an important role for the germination of seed. So these seeds, they will absorb or they will take oxygen from the atmosphere. Okay, from the atmosphere they will take oxygen. Okay, they will take oxygen, and that oxygen they will use for their breathing. We call it as respiration. They do the respiration process to produce energy, and that energy is utilized for the further development of the seed. Okay, the third we can call it as temperature. Temperature, water, oxygen or air also you can write, and temperature. That should be optimum temperature for the development of seed. Generally the optimum temperature for the development of seed varies from seed to seed. It is not fixed for a particular seed. So generally 10 degrees Celsius to some 32 degrees Celsius will be the optimum temperature for the germination of seed. Okay, 10 degree to 32 degrees Celsius, it is the optimum. Optimum means suitable temperature for the germination of seed. But remember that optimum temperature is not fixed for all the seed. It varies from seed to seed. Okay, that's why we cannot say that this seed will germinate. This is the only the optimum temperature for the seed germination. It varies from seed to seed. So here we have talked about the three conditions or three essential conditions for the germination of seed. What are that? Water, air or oxygen, and temperature. Okay, so here we have already discussed about what? Germination. So germination is a process in which the dormant seed will develop into a new plant. You can examine, you can find the difference between the germination of a monocot seed and the germination of a dicot seed. Okay, so again go through the topic. In our experiment, I will show you how these things, these conditions are necessary for the development of germination. One small practical experiment we will do, and the same you will repeat by your own, so that you will be very clear about the topic. Okay, so by this we have completed our chapter, or we have completed our part, that is the development of seed from a flower. Okay, if you have remembered the first part, we have discussed about flower, how from flower a fruit is developed, how inside the fruit we got a seed, and how from that seed again a new plant is developed. Okay, this is the whole cyclic process. That again this plant, again it will form a flower, again it will form fruit, again it will form what? Seeds, and from that continues from generation to generation. So this is all about germination. So I will show you this in experimental form, that all you can repeat also by your own. It will be very interesting to do. Okay, so next period we will have a